Hi everyone, welcome to our first DIY research tutorial. I am Mohak and with me I have Nitish, and we are going to take you through these series one by one. And this is a part of our research at home outreach program that we as a group called Bugbears at NCPS Bangalore have started recently. So without further ado, let's dive right into today's topic, which is going to be antibiotics and their mechanism of action. Now, let's say there's this person who gets sick. He goes to the doctor does some diagnosis and finds out that the patient might have a bacterial infection. So it is likely that the doctor would give an antibiotic to combat this infection, which needs to be taken by the patient over a span of few days. Now, antibiotics are small molecules that usually target the central processes in a bacterial cell. Now, these antibiotics could act on the bacteria in two ways. It could either kill the bacteria, and that is what we call as a bactericidal antibiotic, or it could inactivate the bacteria. That is what we call as a bacteriostatic antibiotic. In either of the two cases, the end goal is to clear the bacteria from the patient's body, and such that the patient becomes healthy again. Now, one antibiotic usually targets one mechanism in a bacterial cell, and there exist different antibiotics in the market, so one can think of different targets that these antibiotics can have. So let's look at some of these mechanisms by which antibiotics might work. First is we can talk about antibiotics like beta-lactams or vancomycin. They can target the cell wall of a bacteria. They might target some region in the cell wall such that they might rupture it and the bacteria would die. So this would be a bactericidal action. There can be antibiotics like rifampicin or fluoroquinolones. So for example, rifampicin, they target processes like RNA synthesis, the RNA synthesis machinery. Or you can have fluoroquinolones, which target the processes which are related to DNA synthesis. Now, the take-home message from this slide is that an antibiotic would have a target inside a bacterial cell that it would use to combat the infection. Next, one can also think of another scenario. Let's say the doctor would have given an antibiotic for the bacterial infection, knowing that previously it has been shown that the antibiotic works. But it can totally be possible that the patient can have a bacteria in a way that the antibiotic might not work against this bacteria, and so he remains to be sick. This is what we call as resistant to the antibiotic. So the bacteria is resistant to the antibiotic. And one of the possibility is that the bacteria might harbor a genetic alteration, which helps the bacteria to be resistant to the antibiotic. So the term to note over here is genetic alteration that the bacteria might harbor. Next, let's try to see what the sources for these genetic alterations could be. Now let's imagine that there is a bacterial population that exists in a given environment. Now, owing to small division times and large bacterial population sizes, one can expect mutants to exist in a given bacterial population. So two things to note over here: small division time and large population sizes, which can give rise to mutants that will always exist in a bacterial population. Now, these mutants could arise due to two reasons, broadly speaking. First, it could be an intrinsic source, where mutation or changes in the DNA at the nucleotide or chromosomal level can occur in a random fashion. So that's your intrinsic source, which is mutation. Second source could be an extrinsic source, where the bacteria takes in the genetic material from its surrounding, for example, from other bacteria present in the vicinity of the bacteria. Now, these mutants could be either fit to survive in the environment, and therefore they will remain in the population, or these mutants could be unfit to propagate. Now, you can think of an unfit bacteria, let's say it could have slower growth rate, or it could, if we are talking about a bacteria which causes infection. So an unfit bacteria in a population could have reduced virulence when compared to other bacteria in the population. Now, due to these processes, there will be variation that will always maintained in a bacterial population. And this is what we call as a standing genetic variation. Now, let's come back to our example of antibiotic um, resistance, and let's see how these genetic alterations might come up in a particular bacterial population. So let's imagine that you have a population to begin with some standing genetic variation. We talked about the reasons about that. And these now, in this case, we should focus on the fact that the colors that are given to these bacteria depend on the fact that they might either be susceptible to the antibiotic. So all the bacteria which are in yellow or on the yellow side will be susceptible to the antibiotic. So the antibiotics will work. Or you can have bacteria in the population which are in red or on the red side. These will be resistant to the antibiotic. Now, one thing to note over here, in this initial population, we have still not introduced the antibiotics. So we talked about how the standing genetic variation will can still occur in the population. Now let's put in an antibiotic. So this is a kind of a stress that we are giving, and we are changing the environment. So we are introducing an antibiotic. Now you can also think of other kinds of stresses like pH or high salt condition, which can also act as stress. But let's focus on antibiotic stress for now. So now in the new environment, let's say the ones which were susceptible will no more be fit to exist or survive in the new environment with the antibiotic in place. And therefore they will die because these are susceptible to the antibiotic. But the only bacteria that will remain after antibiotic has been introduced will be the ones which will be in red. Those are the resistant forms of the bacteria. And these will be the ones that will finally propagate and will dominate in the population. Now, given that how resistance can come up, again, let's go back to the fact that antibiotics, they work. So one antibiotic works on a particular mechanism in bacteria. So one can think of how even the antibiotic resistance can be achieved. So you basically have to disrupt the target of an antibiotic. So one of the things that we can think of is what has been seen in the case of antibiotic resistance related to beta-lactams, macrolides, tetracyclines, aminoglycosides, and so on, is that these bacteria harbor these efflux pumps that help these bacteria to throw out the antibiotics, these antibiotics that are mentioned over here. Another scenario one can think of is that the bacteria which might be resistant to an antibiotic might harbor these alterations in their DNA such that they can modify the targets which usually antibiotics like fluoroquinolones or rifampicins or vancomycin would target. So for example, in case of fluoroquinolones, it can target the DNA synthesis machinery, or rifampicin, it can modify the RNA synthesis machinery. So one of the take-home message from this slide, and a good point to end this video also, would be that when we talk about antibiotics and antibiotic resistance, one can think of two scenarios. One is that the bacteria which might be antibiotic resistant could have genes, could harbor genes that would help them to become resistant. So in our case, we talked about a flux pump. So the bacteria might, the susceptible bacteria might not have efflux pumps. So therefore the antibiotics would work. But the resistant bacteria could have efflux pumps. So therefore they would become resistant to antibiotic. Another scenario could be that these bacteria which are resistant could harbor genetic alterations, which makes them resistant to antibiotics. So for example, we talked about fluoroquinolones or rifampicins, where the modifications can occur in the bacteria. So we are going to explore this in the next video as well, where we are going to take a specific case study of antibiotics that Nitish would be taking ahead. And we are then going to see how one can study if a bacteria is resistant to antibiotic or not using computer and other computational approaches. So with that, we would like to take your leave. See you guys in the next video. Till then, be safe. Take care.